A lot of what my group has been working on over about the last decade now has been to develop tools for culturing cells in a way that, if not able to quite replicate in vivo conditions, can at least approximate important attributes. We basically start off with material that's a bit like jello. It's made of gelatin. We put another chemical that can absorb laser light. So when our laser is focused through the gelatin, it causes gelatin molecules to link up with each other. You imagine the laser focus causes a reaction that bonds one gelatin to another, but also to many other gelatin molecules. If you do this many times, you end up converting a gelled precursor into a solid reagent, so that when you warm up the gelatin, imagine with jello, you warm it up, it all melts away. In our case, everything melts away except where our laser has been, where it's formed these permanent linkages to make this complicated three-dimensional shape. So the printing process is controlled using basically the heart of a computer projector. When you plug your computer into a projection system, the picture on the screen of the computer is reproduced on a little chip within the projector, so that a lamp that shines off that chip projects your image onto the screen. We use the same chip, but in our case we scan our laser across the chip, and this directs the production of an object within our gelatin reagent. If we want to produce a three-dimensional structure that has changes as we move up through solution, we basically project one image, and that corresponds to the object in one plane. You think about slicing through an object. We change what picture is being displayed, and we move our laser focus a little bit deeper, and it produces that image. And we can do that over and over as a stack of images that's reproduced then as a three-dimensional object. So the scale of the objects that we make with biological 3D printing is much smaller than with conventional 3D printing. If you think about the size of a hair on your head, and you take about 1% of that, and then you think about a quarter of that, that's approximately the size of a feature that we can make. And the way this is done is by focusing a laser beam down to those scales and scanning that laser focus through the gelatin in 3 dimensions. This 3D printing approach allows us to organize cells into very complex arrangements of populations. We call them communities, and to evaluate how different populations in a community on a microscopic scale interact. This provides us with potentially very important insights into how polymicrobial communities that exist, for example, in chronic wounds or in a lung that's been infected with bacteria, interact, and how the properties of the bacteria may change to these differences in geometries. By being able to organize cells in ways that may replicate at least certain attributes of how they exist in vivo, we hope to be able to answer important questions of the interactions of these cells.